Hey everyone, so this is our last video for this week, week 3 in physiological psychology, and we are going to cover action potential. So in our last video, we covered resting potential of neurons. Again, on average, which again, it can vary from neuron to neuron, but the average resting potential or electrical charge for a resting potential is negative 70 millivolts. And that's going to be helpful when we start looking at some of this information for action potentials. Action potentials are messages sent by axons which will disrupt the resting potential. Hyperpolarization is when there is increased polarization due to an increased negative charge inside the neuron. So remember, going back to that negative 70 millivolts, it's already negatively charged inside that neuron. So if we add any more negative charge, that is going to make it even more negative and therefore hyperpolarized. Depolarization is when we are reducing polarization towards zero, meaning that instead of being negatively polarized, which again around negative 70 is our average, it's going to be moving towards zero, which will make a little more sense when I blow up these graphs of different electrical charges and what that might look like visually. But before we do that, let's review these concepts of threshold and subthreshold stimulation. So there's this concept of threshold of excitation. And what that means is that each neuron, which are going to be different, are going to have a threshold of excitation, meaning a threshold that is either going to trigger or not trigger a massive depolarization where sodium channels or gates, which we referenced in an earlier video, are opened up that allow sodium to flow in and basically allow that charge to become depolarized and actually into the positive level. And then we've got subthreshold stimulation. So what that means is there might be a stimulation or a signal, but it's not quite enough of a signal in order to reach that threshold where we actually have a larger response. That being said, there is an all or nothing law that applies to this concept, which means that amplitude and velocity are independent of intensity. So either the threshold is met or it's not. And in our text, and you can also do an internet search for this, but each neuron is different in terms of what that threshold might be. The average might be between negative 50 and negative 55 millivolts is the average threshold that a neuron might need to be excited or to have that threshold of excitation met. But regardless, whatever that neuron's threshold is, it's either met or it's not. And if it's over the threshold, it doesn't really matter. So I like the example that the textbook gives of either turning on a light switch or not. It doesn't really matter if you push really hard to turn the light switch on or if you just flip it up. Just as long as you've done it, as you've done that behavior to turn on the light switch, the lights will turn on. So it's the same concept here with this threshold of excitation, is that once that threshold's met, it doesn't matter if the stimulation was more or not. It doesn't make that response any stronger. It's just either met or it's not for this particular concept. So let's take a look at, let's say we were measuring someone's brain activity and this is what we saw. This is an example of hyperpolarization, which is an increase in polarization. So again, remember, around negative 70 millivolts is where our resting potential is normally averaging to be. And so this is an example of that hyperpolarization even becoming even more negative. And then as you can see, it sort of quickly evens out. These are examples of subthreshold stimulations. So our neuron gets a little stimulation, a little electrical charge, and in this case, it went up by 5 millivolts. And in this case, it went up 10. And normally when it's subthreshold stimulation, that response electrically is going to be small and it's going to quickly decay. So as you can see, this is our time axis right here. it's not lasting very long. And this is an example of a stimulation that met that threshold of excitation. So again, it's a massive depolarization. And if you think back to that video where we saw the membrane of the neuron and those sodium channels that allow sodium in or out, when the threshold of excitation has been met, those sodium channels will open up and allow that sodium to come in. And in this case, this electrical charge went up to a positive 30 millivolts, and then it kind of went back down. And we're going to talk about in the next slide this idea of a refractory period, and just sort of how there's differences in this line, because you can kind of see how it even almost goes down to hyperpolarized, and then it sort of evens out again. The peak of an action potential will vary among axons. And so what that means is that we can average out, just kind of like the -70 number, we can average out what the threshold is, what we think it might be on average, or what the average resting potential is, and what the peak might be for certain neurons. But it's really going to vary among different neurons and their axons. Let's review again what we're talking about. So this is sort of a step-by-step molecular basis of an action potential. So the neuron is starting off at a resting potential. And when the neuron is at a resting potential, sodium ions are mostly outside of the neuron and potassium ions are mostly inside. The electrical and concentration gradients are nearly in balance. So remember, we've got that sodium potassium pump that's sort of monitoring what's going on as far as the ions, and then it's trying to maintain that general -70 millivolt resting potential. So that's where we're starting from. And then if that neuron gets a stimulation or an electrical signal, that membrane can become depolarized. And if it reaches the threshold of excitation, those sodium and potassium channels in the membrane are going to open up, and the electrical and concentration gradients are going to drive sodium into the neuron into a reverse polarity. So reverse meaning it's going to go from negative to positive. At the peak of the action potential, the sodium channels will close. Try to visually sort of understand what's going on here. So let's take a look at this figure that helps us put a visual to what we're talking about here. So depolarization, which again is an electrical change within a neuron from a relatively negative charge or resting potential to a positive charge. So this is a picture that shows us that resting potential, we're at the -70 millivolts roughly, and you can see we've got that sodium, potassium in there, and then we've got more sodium outside of that neuron than inside, and more potassium inside than outside. And at this point in the resting potential, that sodium potassium pump is sort of monitoring the concentration levels, helping keep that at the levels that it needs to be at. But then if we receive that, the neuron receives that stimulus, especially, a stimulation that is above the threshold of excitation, that will change. And as you can see here, it's showing that sodium channel, which was ordinarily closed before it became stimulated, is going to allow that sodium to come in. And as you can see, there's more sodium now in here than there was, which is going to create electrically a change. So if we look at it back from that point of view of looking at a table where this is millivolts and this is time, basically this is what we would be seeing again. Resting potential here, which is number one. As you can see, resting potential is here. And we've got that sodium potassium pump just sort of monitoring what's going on and making sure our concentration levels are where they're supposed to be. And then that stimulation comes in, which for this particular neuron example, it's a -55 millivolt stimulation. That's the threshold for this particular neuron example. If that threshold is met, then those gates are going to open up and depolarization is going to happen. So as you can see in this example, that sodium is flooding in, and as that sodium is flooding in, that electrical charge is going up. So in this example, it went up to a little less than positive 30 millivolts. And then in the third step here, it's describing repolarization. And so this is when the sodium channels close, the potassium channel remains open, and it even works towards a temporary hyperpolarization. So a voltage gate potassium channel, which I'm going to describe a little bit more in just a moment, is also helping with that repolarization. So it's basically, it's going down and it's trying to even out, but before it evens out, it's even going to have to hyperpolarize, which again, that means it's going to become even more negative. So you can see that resting potential was negative 70, and it does even out eventually, but before it evens out, it has to get even more repolarized or more negative before it actually evens out. And then number 4, it's then again back to that resting potential where the sodium potassium pump will go ahead and restore the original resting state potential. So propagation of the action potential. So what this particular part of the video is going to cover is how the potential is moving through the axon, okay? So it's the transmission of the action potential down an axon. So it starts in the axon and propagates without loss from start to finish. That being said, an action potential is going to send a signal to the next neuron that it needs to be sent to. So with that being said, in order to get into the neuron, it does have to pass through the cell body, the dendrites, and that is considered back propagation. So it's when it actually is entering the next neuron that it needs to enter into, it's passively registering that action potential through those parts of the neuron. So those parts of the neuron, the cell body and the dendrites, aren't really doing anything with the action potential other than just kind of letting it go through. Conduction velocity ranges from 1 meter per second to 10 meters per second. And the myelin sheath, which you may remember from previous videos, is helping speed up that transmission. So it's helping that electrical charge, that action potential, to move even more quickly than if we didn't have myelin sheath. Saltatory conduction is referring to action potentials hopping from node to node. I'm going to blow up these images in just a moment so we can take a look at that, but you may remember the node of Rambier from previous videos. So you can kind of think of it as that electrical signal sort of bouncing across those nodes. And the refractory period is the time period after the peak of the action potential in which the neuron resists production of more action potential. So what that means is a neuron can't just be doing an action potential after action potential, just one right after the other. There has to be a quick refractory period where it can basically gather itself, get back to that resting potential, and then receive another signal. So as far as the refractory period goes, the textbook identifies that there's two levels of that, basically. We have absolute refractory period, where the neuron is basically like, nope, no way, I cannot produce another action potential at this time. And that particular part of the refractory period will last about 1 millisecond. Then the second level of that is a relative refractory period, where the neuron is saying, no, I'm not going to have another action potential unless I receive a stimulation that is stronger than usual, and that's going to kind of push me over to do it again, even though I may not quite be ready. And that relative refractory period lasts about 2 to 4 milliseconds. Okay, so let's take a look at these pictures to help this click a little bit. And this part of this figure just highlights for us again those different steps of depolarization to repolarization and then again to resting. So in response to a signal or a stimulation, the soma end of the axon, which would be up here by the cell body, becomes depolarized, and the depolarization spreads down the axon. So again, this way, the first part of the membrane repolarizes because the sodium channels are inactivated and additional potassium channels have opened. The membrane cannot depolarize again. So what that means is that it's moving this way, and as it's moving and the signal is going down, the sodium channels immediately are closing after that part of the axon has basically done its job to get it moving. The action potential continues to travel down to the axon terminal. And like I was saying earlier, there's most likely another neuron down here ready to receive that signal. And so it's basically going to be continuing to move that way. I wanted to include this image in here. This is actually in our textbook for saltatory conduction. It just basically shows us that, this is the myelin sheath and those nodes of Ranvier. It basically shows us that saltatory kind of meaning that it's bouncing. It's not just kind of like one straight shot. It's sort of the way that we've got these nodes in between the myelin sheath. You can think of it more as sort of bouncing. In other words, an action potential at the node triggers flow of current to the next node where the membrane regenerates the action potential. So again, it's sort of bouncing from node to node. The last thing that I wanted to be able to cover in this video is sort of an exception to everything we just talked about, and it's called graded potentials. And what this is referencing is the potentials that local neurons will have. What local neurons are smaller neurons without an axon that exchange information with neighboring neurons, basically. So for these small local neurons that are more difficult to study, so we don't quite fully understand all of them yet, they're not going to have that all or nothing law that applies to them. They actually aren't even going to have, in the same sense, an action potential. They are going to have something called graded potentials. And so what that means is that the voltage changes in graded potentials, and so it kind of is going to radiate throughout that particular neuron. And in these cases, the charge is quickly lost through the permeable plasma membrane. And depending on the actual local neuron that we're talking about, it can definitely vary in magnitude in proportion to the intensity of the stimulus. So for these particular types of neurons, local neurons that don't have an axon, I just wanted y'all to be aware that it's basically an entirely different concept of graded potentials. All right, so that basically, in a nutshell, covers action potential and all the components that our textbook highlights for us. This concludes all the videos for week 3 of our class. If you have any questions about anything, I know a lot of these concepts can be very confusing. Please remember that I have office hours and your TA has office hours. And please feel free to stop in, ask questions. And if our office hours don't work for you, feel free to send either of us an email, and we'll do our best to help you at the time that works for you. Thank you so much, and I hope you have a great rest of your week.